Most people are here, so let's start. First of all, let me remind you that there is no class on Monday. It's a holiday, so don't log in to a Zoom session. Second point, on Wednesday, a week from today, is quiz 3. So that will be available to you all day on Wednesday. And our class time will be 60 minutes instead of 75. Test 1, the first comprehensive test, will be on Monday, February 28. So write that down so that you can plan for it. And that will be an entire class period. It will be designed to consume approximately 75 minutes of your time. So there will be no class meeting, no Zoom session on Monday, February 28. Any questions about what's coming up?
Yes, may you please repeat what you said about the first test?
Okay. The first test, so this is a comprehensive test, is on Monday, February 28. So there'll be no Zoom session on Monday, February 28. You'll have all day on that day to take the test. And there will be 2 attempts. So you can log in and take it, and then log in again, take it a second time, if you wish. Okay.
Thank you.
Any question about that? Anything else?
I don't know if I missed it in the beginning when you were talking, but our class is supposed to go back onto campus the 22nd. That's what I heard from the lab professor.
I don't know. I need to find out about that. I haven't heard anything about it. And I don't know if it's mandatory. I'd be curious to know what you think. Would you prefer to remain online or go to campus and meet face to face?
Online for me.
Excuse me?
Online.
You'd prefer online? Anyone else have a preference?
I guess just for safety concerns, I would prefer online as well.
Okay, that's 2 for online. Anybody else want to vote?
Yes, I feel like online's better as well.
Okay. Well, I don't think there's anything that prevents us from staying online, even if the campus is opened up to all courses. So I'm comfortable with that. I don't like being online. I'd like to meet you face to face, but since we've started off the semester this way, it seems to me disruptive to suddenly switch and start meeting in class. So unless I hear differently, let's just plan to keep having online Zoom sessions. If you have any second thoughts about that, send me an email. Now, I haven't mentioned quiz 2. I mentioned quiz 3 is coming up a week from today. Quiz 2, I was quite happy. You all did well. The average for the quiz was 85%, which is good. The highest grade was 100%. So most of you got most things right, but I'd like to quickly take some time and just run over the quiz to make sure everybody understands. Can you see quiz 2? Cell structure and
Yeah.
function? Okay. So there were only 7 questions. The first one was multiple answers. The components of the cell theory. I think everybody got this all right, so I'll go over it. The second one is matching. What kind of microscope would be best suited? Wouldn't mean it doesn't mean it's the only one, but best suited to observe a plant cell. Be a compound light microscope, bacterial ribosomes, transmission, fruit fly antennae. Now my preference here would be a scanning electron microscope because I asked for surface features, but I accepted people who said dissecting microscope or a hand lens. Virus internal structure would be transmission, and living amoebae would be a compound light microscope. The emphasis here would be on living. You can only observe living specimens with light microscopes. Electron microscopes, the specimens have to be killed and fixed in some kind of a medium in order to observe them. So living amoebae, compound light microscope, or you might even use a dissecting microscope, which I would give you credit for. Third question. Genes of eukaryotes are in the nucleus, and prokaryotes, they're in the nucleoid. The Goldilocks conditions, I've listed some here, and the one that's missing is water. Then I ask you to write a short phrase, 2 short phrases for why cells are small. There are 3 main possibilities: transport and communication, cell specialization, and injury repair are all reasons why cells are always small. The next one is matching. And I think everybody got all of these right, so I won't belabor it. And finally, what characteristics are unique to prokaryotes? Well, they would be nucleoid, flagella, no membrane-bound organelles, and a spiral shape. So I'd like to congratulate you for knowing this material in general. You all did very well. And I'm looking forward to quiz 3 coming up in a week. Any questions about quiz 2?
Yeah, I have a question on number 7. So for the ones that are uniquely characteristics of prokaryotes, I didn't put flagella. I thought that some eukaryotes did have flagella.
Do you have any in mind? I can't think of any offhand. I'd have to look that up.
No, I don't have any in particular. It's just like previous knowledge. I just assumed, but I thought, but I mean, again, I'm probably mistaken. I just wanted to double check.
Well, you might have seen or heard that somewhere. So let me check on it. And if I can find evidence that eukaryotes can have flagella, I will go back and give you credit for that.
Okay, sounds good. Thank you.
Okay, anything else? All right. You have a worksheet that was due at noon today. Most of you did it. A couple of you haven't done this yet. And so if you do it late, I will take some points off for being late. But let's go over the questions for that. Should be able to see a colorful grid. Let me know if you can't. Make this bigger. So this is chemistry for biology. Most of you got most of it right. I'd like to just focus on the ones that some people had problems with. The first such one is in the gray box, what type of formula would C6H12O6 represent? Quite a number of you said glucose, but the question asks not for the name of the compound, but for the formula, and that would be molecular. The next one beside it, describe how an ionic bond is formed. Most of you got this either completely right or partially. There were two parts I wanted to see you respond with. One is that one atom loses an electron and another atom gains it, and that causes ions of opposite charge. And so the second point is that those ions attract, and it's that electrical attraction that forms the ionic bond. And the next one over on the left is water polar or non-polar. I think you all got it right. It's polar, and most of you explained it properly. It said electrons are skewed to one side of the molecule. Then all the rest of these, I think everybody got them all right. So most of you did very well on this grid. There's another one, another assignment coming up on atomic structure. That is due, I think, next week. So we'll go over that after you've done it and submit. I don't think anyone has submitted that yet. So any questions about this worksheet? Topic of the day, biomolecules. There are 4 types: nucleic acids, proteins, carbohydrates, and lipids. We'll go through these one at a time and discuss their characteristics. But before doing that, let's recap what we've done so far. We've talked about cell structure and function. The thing to remember here is that the range of sizes, so we've gone all the way from tiny things that we can't see, atoms, to molecules, cells, organisms, planets, and we talked about the universe, galaxies in the universe. Then we talked about how we classify organisms. How do we make order out of this enormous number of different types of living things? So we talked about naming the unique binomials, how phylogenies are produced today, not by just looking at things and comparing morphologies, but by molecular means, sequencing ribosomal RNA molecules. And we talked about the origin of structures, some are analogous, some are homologous. And then finally, just prior to macromolecules, we talked about the chemistry of any molecules, starting with the structures of atoms, the periodic table, and valence electrons, and the relevance of valence electrons to the formation of bonds. Bonds are necessary to make molecules. And we considered, why do bonds form? They don't just happen. Bonds form for particular reasons, and you need to know what those reasons are. Various types of bonds, you just had that on the worksheet. And then we talked a lot about why out of 95 elements in the periodic table, only 4 of them are predominant in living things. So what is there about these 4 elements that make them so important for life? And finally, hydrogen bonds. Hydrogen bonds are everywhere. They're essential for life. They're essential for a number of different processes. They determine the environment of the planet. We talked about the uniqueness of water. It's the only substance on the planet that has that set of characteristics that it has. And we talked about adhesion and cohesion with respect to water. So when you study, think about the recap here of what we've done so far, and try to make it all make sense in your mind, and you'll be able to answer any questions. So we can transition now from the chemistry of life to biomolecules, or molecules that are found only in living things. And they come as either single units called monomers, or multiple units joined together called polymers. So a monomer would be a nucleotide, the polymer would be DNA or RNA. So monomer is an amino acid, the polymer would be a protein. It's a monosaccharide, the polymer would be polysaccharides. And finally, if a monomer is fatty acids, the polymer would be lipids. We're going to talk about all these now. Starting with nucleic acids, 2 types, DNA and RNA. So DNA makes up the genome that stores a genetic code. RNA takes information from that code and makes proteins. But let's look carefully then at the structures of DNA and RNA and compare them. Now, what you see pictured here is a sequence of the units of DNA or RNA. And these units are called nucleotides. Each nucleotide has 3 components. In yellow, you see the phosphate group. Blue, you see the sugar. And in gray, you see a base. These units are connected through the phosphate group and the sugar group. And we'll talk in a little bit about how these groups are connected. But what's important here for you to know at this point is that the phosphate and sugar groups are repeating. It's this monotonous long chain of nothing but phosphate, sugar, phosphate, sugar, phosphate, sugar. And what distinguishes nucleotides are the bases. So there are 4 different bases in DNA: thymine, adenine, cytosine, and guanine. And that's all there is in DNA. Nothing but a chain of sugar phosphates and individual nucleotides distinguished by one of 4 bases. A DNA is a double helix, which means that it's wrapped around two helical chains wrapped around a central axis. And you can see a diagram on the left and then a space filling model on the right. And what you can notice, let's go here. We'll compare DNA with RNA. So DNA, a nucleotide is, as I said, it's a phosphate, a sugar, and a base, in this case, thymine. And DNA is a double-stranded helix. So you see the sugar phosphate backbone. It's nothing but sugar, phosphate, sugar, phosphate, sugar, phosphate, forever. The bases then are unique to particular nucleotides. And in DNA, these bases are joined to each other by hydrogen bonds. And when we talk about how DNA replicates itself, you'll see how important it is that these are hydrogen bonds. The A and T are held together by hydrogen bonds. G and C similarly. Now, if you look at RNA, you'll see there are no hydrogen bonds because it's single-stranded. So the bases would have nothing to hydrogen bond to. So that's important to remember when we look at the differences between DNA and RNA. DNA is double-stranded, it has hydrogen bonds. RNA is single-stranded and it doesn't. DNA contains thymine as one of its bases. RNA contains uracil instead of thymine. RNA molecules are relatively short. DNA molecules are long. We've talked about the hydrogen bonds. And a point I want to make here is that DNA and RNA both have many covalent bonds in addition to the hydrogen bonds. So the entire structure is held together by covalent bonds. The entire structure of a nucleotide. All of these bonds are covalent. Only the bonds between the bases are hydrogen bonds. And this gives you a close-up look at how the hydrogen bonding occurs. So first of all, let's remember what is a hydrogen bond. It's an electrostatic attraction. And it always involves a hydrogen atom in one polar molecule bonded to an electronegative atom in another polar molecule. And it's important in terms of living things, important to remember that oxygen and nitrogen are electronegative. So if you look at the hydrogen bonding between A and T, here's a hydrogen atom. And it's bonded to an oxygen atom, electronegative. Here is a hydrogen atom bonded to a nitrogen atom. And there are 2 between A and T. In the case of G and C, guanine and cytosine, there are 3. But always it's a hydrogen atom bonded to an electronegative oxygen or an electronegative nitrogen. Here, and as I said, G's and C's have 3 hydrogen bonds. So they're a little bit stronger than A and T. So any questions about what a hydrogen bond is and why they form between A's and T's and G's and C's? So let's turn to proteins. What is a protein? It's a chain of amino acids. It's a long string of amino acids. You can see represented here at the bottom of this. And the amino acids are joined by what are called peptide bonds. And they are formed by a dehydration reaction. And dehydration means a loss of water. So you have two amino acids, one with a carboxyl sticking out, the other with an amino group sticking out. There is an H on one, a hydroxyl on the other. Between the two becomes H2O, which is eliminated, and the two amino acids are joined, which you can see in the middle here. So I want to emphasize what is dehydration synthesis because it will come up over and over. If you have two molecules, which I just showed you as amino acids, and water is eliminated, you get a bond formed between the two molecules and water is lost. The opposite of that reaction is called hydrolysis, where you have two atoms or two molecules hooked together. You add water and you get individual units. So this is called hydrolysis. The removal of water to join two molecules is called dehydration. So let's focus then on what an amino acid is. All amino acids have this group, these groups shown here on the left. All amino acids have that. An amino group, and a carboxyl group, and a hydrogen atom, and a side chain represented by R. If you look on the right, there are 20 amino acids found in living things, and they all have this structure, amino group, carboxyl group, and then a side chain, which makes the amino acid unique. Now I want to focus in on peptide bond formation. So the joining of two amino acids is by a peptide bond. Now we've just emphasized that you have a carboxyl end on every amino acid. You have an amino end, and if those face each other, a water molecule can be removed, which then joins this carbon with this nitrogen. You see that here in the blue box. This bond between this carbon and this nitrogen, I've circled it in red, that is the peptide bond. But,
Professor,
yeah.
sorry, I had a question on the amino and carboxyl. Is the carboxyl group, is it always going to have the central element of carbon, and the amino is always going to have that central element of nitrogen?
Yes.
Okay.
Yeah.
That's easier to remember.
Yeah. It is. And if you just keep these in mind, you have a worksheet coming up where you'll draw these these structures. But you're exactly right that amino groups are always NH2, carboxyl groups are always C double bond OH. But now I want to distinguish a peptide bond. As I just said, you have this dehydration reaction, which joins this C with this N, and that is a peptide bond. Now, any other bond between C and N, so for example, here you have a C and an N, that's not a peptide bond. Why? Because to be a peptide bond, the context has to be C double bond O, N bond H. So is that clear? That there are CN bonds of more than one type, and it's only in the context within this blue box that it's a peptide bond. So on the left, I've tried to emphasize that a peptide bond is C bond N, but only if it's flanked by C double bond O and NH. All other CN bonds are not peptide bonds. And here you see a peptide bond in a space filling model where, again, you can see that the peptide bond is a C that's linked to an oxygen and an N that's linked to a hydrogen. Any questions about peptide bonds? This will come up again. Now, there are 20 amino acids found in living things, and here they all are. And what I want to re-emphasize is that they all have the same structure except for the side chain. And we don't have to talk about the structures of each of these. You don't have to memorize them. One thing I would like to point out is that there are two amino acids, methionine and cysteine, which have an atom of sulfur. There and there. This will come up again when we talk about folding, because these sulfurs are very important in the folding of proteins so that they can have activity. For your information, I've listed the structural functional groups found in amino acids and compared them with a lot of other functional groups that are found in living things. So an amino group is found in amino acids, a carboxyl group is, and they're distinguishable from all the other functional groups. We'll talk a lot about phosphate groups when we talk about DNA and RNA. Amino acids and human health. So we have 20 amino acids. We can make 10 of these ourselves. We're capable of synthesizing 10 of the essential amino acids, but the other 10 we get from the food we eat. And the 10, I won't go through them, but the 10 that we need to eat are listed here. So the proteins that have all the essential amino acids are called complete proteins. The others are incomplete, and these are found in plants. But don't worry. So for example, if you look at rice, wheat, and oats, they're all missing the amino acid lysine. Beans are missing methionine and tryptophan. Peas and corn are each missing something. But don't worry, if you're a vegetarian or a vegan, you can eat only plant materials and still get all the amino acids you need by complementation. So for example, if you had a diet containing rice and beans, it would have a complete set of amino acids because beans can complement rice and wheat with lysine, and rice and wheat can complement beans with methionine and tryptophan. So much for amino acids and proteins. We'll switch now to carbohydrates. Carbohydrate means hydrate of carbon. They always have hydrogen and carbon and oxygen in a ratio of 1:2 to 1. So remember, we talked about empirical formulas. If you boil them down to their common denominator, the empirical formula for carbohydrates is CH2O. Carbohydrates, or I should say sugars, names of sugars almost always end in OSE, OS. Glucose, sucrose, ribose, and almost any other sugar. And it's important that you remember that, so that if you see a word ending in os, you're almost certain that it's a sugar. In contrast, when we get to it and talk about enzymes, enzyme names always end in ASE, ACE. So if you see a name ending in ASE, it's probably an enzyme. So keep that in mind. OS is for sugars, ACE is for enzymes. Monosaccharides are monomer units, single sugars. So for example, glucose is a monosaccharide. They're very soluble and they're important substrates for building other molecules. And they're often parts of macromolecules like DNA. Well, most sugars have alternative structures. So if we just consider glucose, it can be, when it's in solution, can be either linear or cyclic. These are structural formulas. Why? Because they show the bonds between the atoms. And you can have a structural formula of more than one type. So here is a cyclic structure of glucose, and here it is abbreviated. Now, in solution, about 3% of glucose molecules are linear, like this, but it's still glucose. So at equilibrium, it's about 97% cyclic and 3% linear. Now, there are isomers of sugars, which are different. They have different names and different features. So glucose, fructose, and galactose are isomers. They all have the same chemical formula, C6H12O6, but they're rearranged. So in glucose, the carboxyl is on the end. In fructose, there's a carbonyl within the chain. And this is actually significant because fructose is sweeter than glucose. So just by changing the position of this functional group, you change the taste. When you look at labels of processed foods, you'll often see that something's been added called high fructose corn syrup. Well, why would the producer use fructose instead of glucose? Because fructose is sweeter, and therefore you use less of it than you would glucose or some other sugar. Disaccharides are two monosaccharides linked together. Here are some examples: maltose, lactose, and sucrose. You take two glucose molecules, do a dehydration reaction. This is dehydration synthesis. Now those two glucose molecules are linked, and it's called maltose. And the water is a byproduct of the reaction. Then you can have much longer chains, longer than disaccharides. And these are largely storage molecules. And all of these, the ones you see here, starch, glycogen, and cellulose, they're all composed entirely of glucose monomers. So what distinguishes them? How are they different? Well, by their linkages and their branching patterns. So starch has long chains of glucose, both branched and unbranched. with these linkages. I'll talk about the linkages in a little bit. Glycogen is the same, except its chains are relatively short and they're highly branched. Whereas cellulose has long unbranched chains that are hydrogen bonded to each other. The chains are hydrogen bonded to each other, which you see the little dots here shows the hydrogen bonding. Sugar linkages. So on the previous picture, I mentioned alpha 1,4 and alpha 1,6 linkages. Well, what does that mean? So alpha linkage versus beta linkage. So if we look at alpha glucose compared to beta glucose, in the case on the number 1 carbon, and you'll have to remember that carbons are numbered starting here, so that there's an oxygen in the ring here. Next to that, the first carbon is 1, 2, 3. Here's carbon 4. Here's carbon 5 and carbon 6. Now, if the hydroxyl is below the plane on carbon 1, it's alpha. If it's above the plane on carbon 1, it's beta. So that's what distinguishes an alpha linkage from a beta linkage. Now, what about 1,4 versus 1,6? As I just said, you number the carbons, 1, 2, 3, 4, 5, 6. So a 1,4 linkage is when the number 1 carbon on 1 glucose is linked to the number 4 carbon on the adjacent one. So that's a 1,4 linkage, and since it's below the plane, this is an alpha 1,4. If the linkage is between the number 1 carbon and number 6, which is this one, it's called a 1,6 linkage. And in this case, again, the oxygen is below the plane on this atom, and so it is alpha 1,6. So is that clear? Anybody have a question about alpha versus beta or 1,4 versus 1,6?
Can you just go over it one more time? I just got really confused.
Okay, so let's back up one slide and remember that these polymers, starch, glycogen, and cellulose, are distinguished from each other by the amount of branching they have and the linkages, alpha 1,4 and alpha 1,6 linkages. Well, what does alpha 1,4 and alpha 1,6 mean? And that's what I'm explaining here. So first, let's separate them and talk about what's alpha versus what's beta. The carbons are numbered from 1. If you start with the oxygen and then go to the right of the oxygen, the first carbon there is number 1. And then you go around, number 2, 3, the one at the end is 4. Next one is 5, and then the branch is carbon 6. If there's a hydroxyl, as there is in glucose, there's a hydroxyl on the number 1 carbon, and if it's below the plane of the carbon, it's called alpha. And it's represented by just being below the 1. So this is an alpha glucose, because the hydroxyl is here. Now, if on the other hand, it's above this plane, it's called beta. So that's what distinguishes an alpha configuration from a beta. And then we have the issue of 1,4 and 1,6. What does that mean? Well, it goes back to the numbering of the carbons. Remember, if you start with the oxygen, immediately to the right is carbon 1, 2, 3, 4. And here, the 4 carbon is linked through an alpha linkage to the 1 carbon of the adjacent molecule. So this is a 1,4 linkage. And all of these linkages are 1,4 because it's always the number 1 carbon linked to the number 4 carbon. And these are all the oxygens are below the plane. So this would be alpha. Now, in some cases, to get branching to occur, you have a 1,6 linkage. So in this case, in the case of branching, the number 1 carbon, shown here, is linked to the number 6 carbon of the adjacent molecule. And since the oxygen here is below the plane, it's alpha. So this is an alpha 1,6 linkage.
Thank you. That makes more sense.
Okay. Any other questions about these linkages? I go into this in detail because if we say that these polymers, starch, glycogen, and cellulose, they're different, but they're all made entirely of glucose, then how do you tell them apart? Well, you tell them apart by the linkages. And this re-illustrates what I've just been saying. Starch found in potatoes, for example, glycogen found in livers, cellulose found in other parts of plants. And remember, cellulose is these unbranched, long unbranched molecules. They're held together by hydrogen bonds. The other 2 types of polymer are all highly branched. The starch is much longer chains than glycogen. And now we can switch to lipids. Lipids contain mostly carbon and hydrogen linked together by nonpolar covalent bonds. They don't mix well with water because they are polar, whereas water is polar and these are nonpolar. So they're hydrophobic. They're usually greasy and oily. Now, what are the functions of lipids in living things? Well, they're storage molecules, for one thing. They're components of membranes. And I've circled this function in red because we're going to talk a lot more about membranes. And so lipids are essential for the structures of membranes, and without membranes, there would be no cells. So starting with the smallest or most primitive cells to the most complex ones, they all require membranes to keep their insides separate from outside. The types of lipids. There are 5 types: fatty acids, triglycerides, phospholipids, steroids, and waxes. I've just talked about the membrane components. Fatty acids we'll talk about, and fatty acids can make triglycerides, and these are fats. And there are 2 types which you're familiar with in your everyday life. So for example, bacon fat, lard, and butter They're all fats and they're solids. Why are they solids? We'll get to that in a minute. Other triglycerides are oils, like corn oil, coconut, olive oil, and oils are liquids. Why are they liquids? We'll see. So we'll start with the building blocks, which are fatty acids, which are acidic fats. So a fatty acid in its simplest form is simply a long chain of carbons with the attendant hydrogens. And at the end of the chain is a carboxyl. So that's what makes it an acid. It's called a fatty for the carbon chain, acid for the carboxyl group. A saturated fatty acid is a rigid molecule. Almost all fats are rigid in their carbon chains. There are no double bonds. Now, some fatty acids have flexible molecules, and these are the oils. So oils are flexible, fats are rigid. And the reason this is flexible is that there's a double bond between two of the carbons. Other than that, it's the same. It still has a carboxyl at the end. Well, what this double bond permits is rotation of the molecule around the bond. So the tail of it can rotate around the other end. And that makes it flexible and causes the substance to be liquid rather than solid. Or you can have polyunsaturated fatty acids where there's more than one double bond. Now, how are these molecules made? Well, to make a fat molecule, you take a fatty acid and connect it to glycerol. Glycerol is a 3-carbon alcohol. And here we come again to dehydration reaction, where the hydroxyl of the fatty acid and a hydrogen from the alcohol go together to eliminate a water molecule, which then hooks this carbon to this oxygen, and you end up with the structure you see below. And if you do that on all 3 of the carbons in glycerol, you have a triglyceride, a fat molecule. And that's important in diet. So some foods most of their fatty acids are saturated. So for example, French fries, what you get in a burger. Whereas in other foods, there's a significant proportion of unsaturated fatty acids, that is, double bonds in the carbon chain. These are the liquids, and you find unsaturated fatty acids in fish and nuts and beans, avocado, olives, and other mostly plant materials. So a diet high in saturated fats contributes to high LDL cholesterol in the blood, and that can contribute to heart disease. So when you choose your foods, it's best if you minimize the ones with saturated fatty acids and try to maximize unsaturated. And now let's get to the real nuts and bolts of living things, which are membranes composed of phospholipids. Here you see a space-filling model of a phospholipid. It's a long carbon chain, nothing but carbons and hydrogens. So it can be either unsaturated or saturated. But then at the end, instead of just a carboxyl, there's a phosphate group. And you know what a phosphate group is from previously. And then another group, a nitrogen containing group, and this can be variable. So we won't emphasize this because it's always there and it can be any number of things. The important point here is that these tails, being hydrocarbons, are nonpolar and therefore hydrophobic. The head of a phospholipid is polar and it's hydrophilic. So this is very unusual for a molecule to be nonpolar at one end and polar at the other. And the reason this is important is that if you put them in the right environment, where you have aqueous and non-aqueous parts, the polar heads will always stick out into the aqueous environment, and the non-polar tails will always stick into the non-aqueous environment. To illustrate it another way, if you poured a mixture of phospholipids into a beaker containing oil and water, first of all, the oil would float on the water. The oil is nonpolar, the water is polar. And so the phospholipids would orient themselves immediately so that their tails, their hydrophobic tails, are in the oil and their hydrophilic heads are in the water. So this feature of phospholipids is the basis for biological membranes, which is important for life because without membranes, there could not be cells. Membranes separate the contents of the cell from the rest of the world. And that's illustrated more graphically here, where you see a section of a membrane. Now, all cells are aqueous. That is, the inside of a cell is aqueous. It's based in water. And cells always exist in aqueous environments. I should say usually exist in aqueous environments. And so the membrane that separates the inside of a cell with the outside is composed of these phospholipid tails connected together, and then the polar heads sticking out into the aqueous environment on either side of the membrane. So that's all I want to say about membranes and phospholipids now, but we'll come back to it later. Now let's consider steroids, one of the other classes of lipids. When you see this 4-carbon ring, it's by definition a steroid. So this is the common subunit of a steroid. How do we encounter steroids? What makes each steroid unique is the side chains. So you see that all 3 of these compounds has the same basic structure, but different side chains. So this is testosterone, a male sex hormone. Estrogen differs by side chains. This is a female sex hormone. And cholesterol, which we have in our membranes, has yet a different set of side chains. Fatty acids can comprise waxes. Waxes are very hydrophobic, completely insoluble, and they function, for example, as coverings of leaves, fruits, feathers, and the wax in your ear canal is composed of fatty acids. And this is how waxes are made. A wax is a condensation between an alcohol and an acid. And again, there's a dehydration reaction. A water molecule comes out so that these two polymers are linked together as an ester. And what you get is what you see at the bottom here, this long carbon chain connected with another long carbon chain by an ester group. Okay, I think, yes, so that's it for the slides. Any questions about anything I've said about biomolecules today? Any additional questions? No? Well, I hope that means it was all clear. Just remind you, no class on Monday, so don't log into Zoom on Monday. And a week from today, we'll have quiz 3, which you can take it anytime during the day on Wednesday. And then during our class period, we'll talk about nucleic acids and transcription and translation. Okay, if there's nothing else, let's end the session.